All right, we're back. Live streaming 2370 meters beneath the sea surface. The Damm hydrothermal vent field. We are sampling about 28 degrees Celsius vent fluid. If you can't see us, refresh your browser. You can submit questions via Twitter. Use the hashtag OASES2013 and SOI Cruises or Joules Deep. We've pumped about probably 2 liters of vent fluid through this filter. We're collecting microbial biomass. By the end of this sample, we'll have collected about 3 liters. Should be upwards of 3 billion individual microbial cells. And that's a way to concentrate biomass rather than carry up multiple liters of vent fluid from the seafloor. We bring up some vent fluid. The isobaric gas-tight chambers that you've seen in action sample about 150 ml of fluid. And most of that goes to geochemistry. The bottles on the super sampler collect about 1 liter. And that goes to both chemistry and microbiology. The other piece of equipment we have on the vehicle today is a slurp chamber, sort of like a mini vacuum cleaner on the seafloor. That's to collect things like shrimp, which we'll be doing later in this dive. And what else do we have in the vehicle? Do we have a bio box on? We have a bio box, which is in the front of the vehicle that you can't see. That's in case we come upon any mussels. We'll scoop them up with a net that we have up in our milk crate at the front of the box. The milk crate is just sort of for miscellaneous bits and pieces that we might need on the seafloor. We also have weights in the milk crate to make sure our ballast of the vehicle stays the same and that we're light enough to get back to the surface. Heavy enough to get down, light enough to get back. So Casey and the team do careful calculations to make sure our payload is correct. Does the vehicle have a CTD on it? So it also has a CTD mounted on the vehicle that's monitoring conductivity, temperature, and depth. And it has a EH sensor, which can look for reducing potential in the water column. When you're in vent fluids, you get a reduction in EH. Transmissometer to measure particle concentrations in the water column. And a transponder that talks back to the USBL pole that's hanging beneath the Falcor. That's how we know where Nereus is, using ultra-short baseline navigation. And then the ship uses Global Positioning System, GPS, to figure out where it is. So we know where the ship is, get a relative relationship of where Nereus is to the ship. We know our depth. We know our heading. So it's a complicated way to navigate yourself on the seafloor, but it works pretty well most of the time. Ultra-short baseline doesn't always work that great in really deep waters, where we've been operating at the Picard hydrothermal vent field, where we'll be going back tomorrow. But we've been here before, and we have some pretty good maps, so we've been able to do a good job of finding spots that we want to go to. What's the time? What time is it? Why isn't there a clock? I can never see. I'm too short. You guys have designed these seats for really tall people. 16:18. So let's go to 16:23 on this, Chip. For the record, I'm not short. The average American woman is 5'5", and I'm 5'5.5". Average. But the very comfy pilot seats are probably built for 5'9". So my feet don't touch the floor when I'm sitting here. Really? Chris got that camera for his 50th birthday. Did you take some silly pictures?
No way it came from, man.
He won't know. Chris German accidentally left his camera laying around, so we're trying to add some fun pictures. So the beautiful video footage you see is being taken by high-def cameras that were donated to Woods Hole Oceanographic by James Cameron and the Deep Sea Challenger team. These were the same cameras and lights that he used on his historic dive to the bottom of the Mariana Trench last year. Solo manned mission. Spent less than 2 hours, I believe, on the seafloor, right? James Cameron's a little more. Jettisoned back to the top surface safely. Became the first person to make a solo mission that deep. Then donated a submarine to Woods Hole Oceanographic, which has a new Center for Marine Robotics and a lot of expertise in deep submergence vehicles. And we immediately took it apart, parts of it apart, and took the cameras and the lights, with of course permission, to see how they would light up our world on Nereus. And we've had to fiddle with them a bit, but I think you can tell from the images and video we're getting some really beautiful views of the seafloor. You know, so Nereus is the size of those really little cars. What are those little cars called? That are, no, they're not called smart cars, right? Nereus is about the size of a smart car. It might even be a little bit bigger. And so once you get past about 200, 250 meters, it is dark. And we need lights, but it's still just like shining a light out into the vast unknown. It's very hard to navigate. So the better the lights, the better it goes. All right, Chip, you want to stop that sample? We're stopping that sample at 1623. And we are going to fill one more bottle here so that we can have plenty of water to play with back in the lab. That's going to be bottle number 5. Super bottle number 5. And he is starting pumping right now, 1623. No, this is a bottle. Bottle number 5. Bottle number 5. Nereus sample 14. And then I get to go eat lunch because we're going to slurp up some shrimp. So if you want to see our vacuum cleaner in action, we're going to be doing that. What we've seen at Von Damm are areas of dense shrimp, like what you're looking at now, and then areas of sparse shrimp, where they're sort of spaced out over the rocks. And so we are going to, in one of my points of view off to the left, we can see those sparse shrimp, and that's what we're going to sample next for Cindy and Max. Interested in the stable isotope and carbon signature of the symbionts in them. And also, of course, working on the genetics and population structure and reproductive history and all sorts of things about these shrimp. You can see there's really, really tiny ones, probably juveniles, and the very large ones, adults. Some of them have orange at the head, and what is that? The big ones are carnivores? The big orange ones are carnivores, but you don't see many orange ones in this field, Chuck. But don't, some of them sometimes have like that green blob and orangish blob near their head. Ah, okay. Cool. Green tea. Wait, Chris, where are you going? You're coming right back? Okay. So at lunchtime we had a watch shift here with the ROV team. Chris Taylor and Jonathan Holland, both from Hui, are sitting with me. Not doing much right now, but keeping an eye on the vehicle. We have 4 camera views, and then we have 1, 2, 3, 4, 5 or 6 screens that help us with navigation, with vehicle status, power, hydraulics. What's that one to the left?
That's the depressor.
Oh, then we have another screen that tells us where the depressor is. So right now, the depressor is at 1,000 meters. So it's 1,300 meters above Nereus, and they're connected by that thin fiber optic cable. And what's that yellow? Is that its bounce?
It's the delta depth.
Okay.
between the two vehicles.
Oh, okay. So the depressor is feeling the motion from the surface ocean, and Nereus is protected from that, which is great. There's still deep ocean currents that we've had a little bit of trouble with in some places. Remember, we're at the bottom. Well, in this case, we're at a topographic high out top of Mount Dent here at the Von Damm hydrothermal vent. But those can make keeping the vehicle in place difficult. The screen below the depressor screen shows the forward-looking sonar, which is mounted on the vehicle, looking for obstacles that we don't want to run into. And then to the right of that, we have the navigation screen, which we were streaming on our YouTube video earlier when we were going through the dark water column, which is pretty boring. So we thought we'd show you how we navigate. Chris German has returned with coffee and tea for those on control, on watch right now. And I'm going to be turning the microphone over to him in just a minute so that I can go have some lunch and he can help direct shrimp sucking. Is this your credit card sitting right here? Is that a good idea to leave that around? All right, Chris, do you want to take over so I can get lunch?
Sure.
Okay, handing it over to Chris.
Okay, so back to whatever comes next. Let's pretend we know what we're doing here. So Cindy has some special recipe lined up for this evening. I'm not sure quite what it is, but whatever it requires. In the ingredients apparently are 12 big shrimp. Not shrimp parts, not chicken legs, not chicken wings, 4 fried chickens. Aretha Franklin will be joining us later on to burst into song. So, let's stow the temperature probe. Thank you, Chris. And we're going to be using the slurp gun as our next primary operation. Actually, what we could do, let's see how fast they show up. Let's get the slurp gun in our hand, and then if people haven't shown up yet, start choosing their preferred shrimp. We can collect some nice high-definition video of that site that we've just been sampling. We probably have some nice video of it for the last half an hour with the nozzle in there, but the unadulterated natural environment is always a nice thing to look at. And Cliff spent about 10-15 minutes earlier today dialing in the camera to make sure it was producing really nice results. Leighton, would you be so kind as to tell me what time we finished collecting the last sample?
I'm not going that far now. We finished at 16:30:24.
Ah, that was the previous sample, I think. Oh no, just got some weird numbers. 16:30, thank you. Okay, no, that's right. Somebody had written 16:53 here, which is still 25 minutes in the future.
Now I need
Let's correct that before we go any further.
the brow camera, the brow light. I think somebody just hit the submarine, man. It's on the alcohol.
That's just the Shinkai bouncing up under the ship. So Cindy Vandover has joined us in the control van. She's ready to hand pick her shrimp. You ready to hand pick your shrimp?
Not quite.
Cindy's just finishing dessert.
Yeah, look, I want to go get one name.
Okay. And then perhaps you could tell us a little bit about the shrimp when you come back, Cindy. I know at the start of this morning she had a request for 10 shrimp, but one of her colleagues from shore has been in touch today. So the request has gone up for 12 shrimp for Cindy today, and then also some more for Max. The time aboard the RV Falcor has just gone 12:30. We're on the same time zone as the east coast of the United States. We have been on the sea floor since around 7:30 this morning. We launched just after 6 o'clock. We've been on the sea floor about 5 hours. We've been pretty conservative with our batteries. We went to some really interesting new sites we'd never seen before at the very northern limit of the Von Damm Mound to start the day. And then about 2 hours ago, we left there, did a long circuit out through the water column round to the east of the Von Damm Mound, and then came back to reoccupy a station we sampled yesterday to the southeast of the main spire. We collected some shrimp yesterday, but not all the shrimp we wanted. And so, we may remember if you were participating in the cruise, you saw we had trouble with our manipulator. Guys did a fantastic job overnight, got the manipulator up and running again. We were back in the water at 6 o'clock this morning, and so we're picking up where we left off. Going to collect some shrimp here for Cindy and Max to finish what we started yesterday, and then go back to doing some more microbial sampling for Julie, which is something else that was left unfinished during yesterday's dive. So, Chris is about ready with the shrimp sampler, and we'll be good to go in a moment.
Okay, this is Cindy Vandover from Duke University. We are at 2368 meters and set up to do some sampling of sparse shrimp. So we've got the slurp pump, and we can just start picking out individuals, anything not in that big clump in the center of that hole. As big a shrimp as you can get makes it easier for us to dissect. We were in the lab this morning with the shrimp that we collected yesterday. We'd kept them in the refrigerator overnight, and we were dissecting them. We've been doing that every night, pulling off the branchiostegite, the piece of the carapace that wraps around the body of the shrimp, and Max would take that and scrape it for bacteria to do lipid analyses. And then I would take pieces of the abdomen for genetic work. But today we had the luxury of some time and already had our key samples. We went back to using a microscope. They skip away, don't they? And there's one. And under a microscope, the big shrimp were really, it was really a lot of fun. We were subsampling tissues for Chip Breyer to do some metal analyses with a microscope X-ray fluorescence setup, so we can see where the metals are located. So I was dissecting hepatopancreas, which turns out to be a lobular structure inside the head cephalothorax region of the shrimp. And I was also getting some control tissue for the abdomen, where I don't expect to see metals. But the hepatopancreas is part of the digestive system and might reasonably be expected to be rich in metals. And while we were doing that, there were the stomachs right in front of us. So we started looking at stomach contents, since part of our interest is in trophic interactions for these shrimp. You're fine just driving on your own, right? You're picking out quite happily. And the gut contents were fascinating. These shrimp are two of the large animals we looked at were clearly eating other shrimp. And we know that from looking at the chitinous parts that were in the stomach contents, and also from, we had one partially digested shrimp, but you could see the little claws still left at the head end. So these are meat eaters, much to my surprise. At least some of them are meat eaters. We had another shrimp that we opened up and had mouth parts underneath that carapace that were covered with bacteria, which is more like what I expected. And indeed, in that stomach, Max said, I bet that stomach's full of bacteria. and it was. So that particular shrimp at that moment of time had been eating bacteria, and a dense amount of bacteria. At least that's our inference. It's not fact yet, but we infer. And then we had some juveniles, which are bright red. And it's hard to see on the video when we have the juveniles, but they show up in our samples. And we dissected that again, Max is interested in the lipid compositions of these shrimp, in particular to see if there's any evidence of photosynthetic carbon entering into their diets. So the baby shrimp are bright orange-red. We went into their bodies and removed the red stuff. We froze them first and could dissect out the red. Turns out the red is in the hepatopancreas, and it looks very lipid-rich in the sense that globules of oily material was associated with those hepatopancreas. So we're starting to learn new bits of information about the trophic relationships of these shrimp and the bacteria that they feed on, and their role as predators in the system as well. How many shrimp do we have? How many shrimp have we collected? 5. I'm glad somebody's counting 5 shrimp. So today we're hoping to collect, how many do you want, Max? 50 for Max and 12 for me. So I'm getting 12. I want myself 10, but I'm collecting for Stanton Belford of Auburn University in the education program there. We're picking up 2 shrimp, and Stanton, if you're listening, if you're interested in these shrimp and education, we will be back here again in August with a program called Nautilus Live. You can find it on our website. We'll be here 19 to 30 August. And we'll have live feeds through the Nautilus Live website, so a different website from the Schmidt Ocean Institute YouTube channel. So that'll be Nautilus Live 19 to 30 August back at the Cayman Vents. And I should be there, at least from the 23rd or 24th of August. I'll hop on the ship and join that cruise. That's with Bob Ballard and the Nautilus team. Wasn't that fun stuff? Could you hear the stuff about the diets of the shrimp, Chris? Chris says he's going to listen to me on YouTube to get the story, but I can just tell you in the galley at dinner. I wouldn't tell him at lunchtime. I wanted to keep it a surprise. Yeah, it's a surprise. So we had thought these shrimp were, it's really perplexing because we think the shrimp that are on the black smoker chimneys or the really hot surfaces are indeed in that environment to put their bacteria, their symbiotic bacteria, into warm fluids. The ones that are eating meat, do they wander away from the warm waters to go get a meat meal and then otherwise live on bacteria? It's very perplexing. Hang out by the pool and go for dinner. Could be. Well, we had on a cruise in the Indian Ocean, where there's a similar type of shrimp, similar shrimp behavior, and it was a lot like this. There'd be really dense swarms of shrimp in the warm water and then these sparse things. And one of the graduate students there was talking about it might be really interesting to model the metabolic demands when they go into the cold water. Their metabolic demands lower, so they're not using so much energy, and they could sit there and digest their meal. Yeah. So I don't know. It's a
like a bear in hibernation.
bear in hibernation.
Readjust myself.
I suppose actually along the top of the spire you don't come through a whole area.
It's okay if I reposition a little?
Yes, absolutely. You may reposition, make it easy for yourself. I don't think there's any particular demand for a specific sparse shrimp, just ones that are out in the periphery. Yeah. So tonight we will do a lot more dissections, or tonight or tomorrow morning. Are we doing a CTD tonight? I asked that already. Are we? You don't need a CTD.
I've got to check the numbers out because there's nothing on them. I hope you do.
So I think those of you who've been watching YouTube regularly, which probably includes, right now it shows we have 18 viewers, and my guess is that half a dozen of those at least are from the Duke Marine Lab and my research lab group. So Bernie Ball, Dr. Sophie Pluvier, and Jamie Wagner are probably watching. Some of the admin staff at the Marine Lab, Katie Wood, might be watching, so hi to you all. We when we dissect these animals, you haven't had a chance to see what I was starting to say. But you haven't had a chance to see how we work in the lab with the dissections and the vials. But Bernie Ball sent me out here with one box, and the box is full of little cryovials, vials that you can stick in a freezer that we use to put snips of tissue. So the routine is to go into each animal and get a little piece of abdominal tissue, chop it up fine, stick it in different preservatives. So I'm using something called the RNA later, and I'm also using a 200 proof ethanol, 2 different preservatives, so 2 snips of tissue. And then I bring back very neat samples, little vials come back. It's very satisfying for those of us who like to be neat and tidy. So I've been working on shrimp like these at hydrothermal vents since probably 1986. And we've found them on the Mid-Atlantic Ridge, swarming shrimp on the Mid-Atlantic Ridge, on the Central Indian Ridge, and now here in Cayman. We think that they navigate with this kind of local scale, that they can sense, they can detect sulfide gradients. So we know they have chemical sensors, chemosensory CD that are sensitive to sulfide. We had a physiologist join us on a cruise who did some electrophysiology and showed the signal response to sulfide chemical cues. But largely, chemical cues in vent environments, man, it's a wide open field. If somebody wants to study chemical cues and figure out how animals navigate down here, that's a door that you could just walk right through and really make some interesting observations and probably do some really fun experiments. I think at one point I did put down some scent bottles, and I put in different types of amino acids to see if I could get them to come to dead animal smells. And I was doing it at a seep site where there were snails, and in fact the snails did love the dead smell. They're what do you call them, necrovores? They eat dead things. But that kind of experiment hasn't been done here to see if shrimp would be attracted to a sulfide cue. It becomes even more interesting when you think about the larvae of these animals. How do the larvae of most of these animals at vents, the shrimp, other invertebrates, the snails, and the tube worms, all have a larval dispersal stage. It goes up into the water column, and you can imagine the vast ocean. The water column is big, right? So dilution, they get dispersed. Well, one would think they could get dispersed very far. Yet we often find that cohorts come back to the vent. So a large number of small individuals all come onto a site at once. And how they stay together, maybe that may happen both by behavior of the larvae and by some of the physical properties. But then how do they actually find a vent site? I think that there's probably some chemical cues that they use to alter the behavior. And the main thing they could do is just sink or swim down. Larvae aren't strong swimmers against currents, but they can sometimes take on behaviors that allow them to drop to the seafloor when they sense something good. So chemical cues from the vent fluids may be attracting the larvae, or perhaps cues from the animals themselves, so that they come in to aggregate. If you smell a shrimp, that's a good habitat. So you would try to make your way to a habitat because there's other shrimp there and you can sense them. You're doing good, Chris. Sometimes outsmarted by a shrimp, huh?
Turn off the sleep.
Sneaking up on him. So, yeah. So Chris was just trying to turn off the slurp so it wouldn't disturb them, and then sneak up on them, but they feel the siphon, the hose. Oh, there we go. Oh. So these shrimp are in ambient temperature water. So that's ambient's about 4 degrees Celsius down here. How much? 4.8 degrees Celsius is ambient. Where are you reading that? Where do you read that? Oh, just from the temperature probe of the water bottles. Right. So 4.8 degrees. See, that's pretty cold. We call them hot springs, but the animals are actually quite chilly water. Yeah. The ambient temperature is 4.8. That's where these sparse shrimp are in 4.8 degrees. Yeah. Yeah. Not much of a hot spring, but even where the shrimp are, 20 degrees. Yeah. Tuberums are really cool. These tuberums that we have are in very cool water. The warm areas here are colder than the surface ocean, and do you know the reason for that?
The hot crack site is about 25 degrees.
Yeah.
Where the shrimp are clustered.
Yeah.
Swimming there isn't as warm as
Right, but why is the ambient temperature so warm? 5 degrees is really warm for the deep sea. No, but 2,500 meters on the East Pacific Rise is 1.2 or 2 degrees C.
sunlight for an extra 1,000 meters. That's mixed in Antarctic bottom water and that.
Antarctic bottom water is the explanation for cold Pacific deep water.
Go down to a 5,000 meter site, it
Yeah.
drops another degree or more.
Okay. So at 5,000 meters, we're at 3.8, 3.9.
If we were out in the North Atlantic where the deep water's just come from the Arctic, it's down to about 2 degrees.
Do you want to explain some
Sure.
water? I'm sure they didn't get all that from me.
So Cindy's just handed over the microphone back to me. She's finished, well, she told you that fascinating story about the shrimp. And we have about 14 shrimp in the bag so far, so we're about a quarter of the distance. So Cindy was just talking about ocean temperatures and was interested to try and compare and contrast the temperatures. We were saying that the temperature here is nearly 5 degrees Celsius, which isn't especially hot by normal standards. You wouldn't want to hang around in water that cold yourself. And if you go another 1,000 meters deeper, then we drop another 1 degree. And the temperature there is just under 4 degrees Celsius. But at 5,000 meters, that's still itself quite warm compared to the open ocean. That more typical out in the larger, wider open ocean, temperatures are more like 2 to 3 degrees Celsius at these kinds of depths. So one of the thoughts is that the Mid-Cayman Rise is a bit of a box canyon. It's 5,000 meter deep water, but there isn't any way, if you're at 5,000 meters, to get out of the Mid-Cayman Rise unless you climb up and over the Rift Valley walls like the depths we're at now. So we have a small parcel of water about 20 miles wide and 100 kilometers long. So 100 kilometers by, actually probably about 20 kilometers, so 2,000 square kilometer trough, 2,000, which is about 2,000 meters deeper than everything else around it. So there is actually scope that the entire volume of the deep water in the Mid-Cayman Rise is being warmed up by the hydrothermal activity, because it doesn't get to exchange with the rest of the outside ocean. The fast-spreading ridges from the Pacific, for example, are more like a mountain range that stands tall, and the plumes that actually rise up of hydrothermal fluid from those ridges then get dispersed all the way across the world's oceans. My next research expedition later this year, incidentally, is one I've been spending, like Cindy was telling you about. She's been studying shrimp since the 1980s, and she and I started our graduate school careers pretty much around the same time. And what I've been studying mostly is, or the other thing apart from locating new hydrothermal vents, because they're really fun to come and look at, what I do for my real job is to actually understand the chemical impact of these hydrothermal systems on global ocean budgets. And in the Pacific, there's a particular site where the hydrothermal plumes spread for about 2,000 to 3,000 kilometers all the way across the Pacific Oceans. They get dispersed and diluted. So clearly you don't see much of a temperature signal from the geothermal warming from the Pacific at any one location, because all the heat can be dispersed over a vast ocean volume. If you watch from space, basically if you came in over Tahiti, which is where my cruise is going to end on Christmas Eve, you would actually hardly be able to see any land at all. All you can see is a large ocean, and that's getting down to about 5,000 meters deep. In the Atlantic, it was a colleague of ours, John Edmund, who was my postdoctoral advisor, used to actually famously say that the Pacific was a real ocean and the Atlantic was just a bowling alley. It's relatively narrow, it's a landlocked ocean basin, although it can take days to get across it compared to the Pacific. It's just, his take was that the cooling of waters just where the ice forms in the Arctic leaves behind very cold and salty dense water as you crystallize out the ice, and it's that water that then sinks and flows south as North Atlantic deep water, and that's what gives rise to the very coldest deep waters that flow along the bottom of the ocean. And that cold water flows down through the Atlantic, turns east around the bottom of South Africa and flows further east into the Indian and Pacific Oceans. And it's joined all around Antarctica. The same process is happening at the South Pole, where freezing of water to form sea ice leaves behind cold, salty, dense water that sinks, spinning off and shedding off the Antarctic continent, or rather the southern hemisphere. And so that's the water that spews out, or basically flows around what we call the thermohaline conveyor, which on average takes about 1,000 years to get from one ocean basin to another.
I'll be down in the lab when you're ready.
That was, you may have just heard the dulcet tones of Julie Huber, letting us know that where I can find her when her sampling station is ready. So we're sampling these shrimp. I've not been keeping track of count. We were at 14, we're up to 19 now. No, I stopped. I'd interrupted my counting at 15, so.
I've got 900, just over 900 meters.
So we're 1/3 distance on the shrimpathon today. So we have 20 shrimp in the bank.
This is Cindy Vandover again, Duke University. Back on the phone. Still sampling shrimp, as Chris just said. Thanks, Chris, for that great lesson in physical oceanography, temperature. And I see our number of viewers has gone up, so I'm guessing that's all the people at the Duke Marine Lab who now called each other and are all watching and the physical oceanographers somehow heard we were talking about that. And also I had mentioned to my brother we were going to be on air this afternoon. So Ken Vandover, hello, from the Caribbean. So for folks who weren't here just a little while ago, might not have heard, we will be back at this site, or I will be back at this site in August, on 19 August to 30 August, with research with the exploration vessel Nautilus. And you can watch our work here live on the Nautilus Live website. So 19 to 30 August, there's a lot of education program that goes along with the Nautilus expeditions. So we should have a good time doing some more shrimp work, but also doing some exploration and moving to some different areas, maybe looking at some of the background fauna. 27 individuals? We're rocking.
Find out some big ones for Christopher.
These all look like the big ones, don't they? But
I don't know.
it's hard to tell.
They should carry little tape measures with them.
Yeah. So big. I am.
Wherever you want.
When we get them in the lab and measure them, I measure every shrimp. We do a total length, and I'm thinking, oh golly, I think the total length on these have been in the 40 millimeter range, 40 to 50 millimeter range. The tiny ones are on 10 millimeters or so, 10 to 20 millimeters. So they're a little smaller than cocktail size
It's
shrimp.
probably about this big.
What's that?
I was just saying we've got a scale
Oh,
in terms of the nozzle of these.
well, I have a mic.
No,
So we
no,
can.
no,
So if you're watching the video,
big ones.
you can look at the that diameter. I think it's about 2 inches. In fact, I know it's 2 inches because I measured it. So for scale, that's a way to tell it. So they're pretty good sized shrimp. There also looks like there's some snails, the Ihea spira, a new species of snail that was described from here by Verity Nye. Is maybe some of those little white dots. We got some of them yesterday when we sampled. They have been preserved for genetics. They're really not fun to work with. Because for genetics, we have to crush the animals and to allow the preservative to penetrate. And crushing these little snails is a hard thing to do. That may be more detail than the audience cares to know about our scientific protocols.
How big the big ones were, Cindy?
I have it on the clipboard. I'm thinking they were in the 40 to 50 millimeter range.
You're right. Then these look as though they're all big.
Well, if they're the same size as
or bigger.
as the opening, they're
Yeah,
2 inches.
that's 50.
Yeah.
Wow.
31 individuals so far, one by one. It's good work.
Except for the two.
No, that was just one. Another one.
Yes, Julie would like 2 individuals. She's going to preserve them in glycerol whole. And we had the 2 from Stanton Belford from Auburn University. Yes, 64 is our target. Halfway there.
Better hurry up, Chris, before the 12 goes up again.
I think your average, what's his average sampling rate? 1 shrimp per, I think he's doing good.
Oh, great.
Yeah. Better than a shrimp a minute. Go, Chris. So I was an Alvin pilot for 2.5 years. I was with the Alvin group, so I used to do this kind of thing myself on the seafloor. And my recollection is you just kind of get into a zone doing this repetitive stuff, and it's kind of relaxing compared to some of the other things you might do down here. Intense, but relaxing. There's no stress, right? You're going to be able to get it, no problem. Sooner or later, just keep going after them. The animals come up.
Another one.
one almost wanted to jump in.
Another one.
38. You're sweeping this place clean. Are you finding if you go to the head end, they come easier?
Come right in on top of it.
On top.
Directly on top.
Okay, so pilot's got a technique going straight down on top of a shrimp, and they don't hop away. So the behavior, or lack of behavior, of these shrimp is interesting. As they sit on these rocks, they really don't do much. See them creeping across surfaces, but
Another
they're
one.
in random orientations, so they're not facing into any current necessarily. They have the classic shrimp escape reaction where they dart backwards fast. So if you've been watching us the past few days, when we talk about the shrimp, one of the things that you'll have heard the story, but for new viewers, the shrimp have on their backs a novel photoreceptor. So these shrimp, first of all we have 3 species of shrimp in this Von Damm vent site. The most abundant one is the one you see here, is Rimicaris hyvaci, aka Corocaris chasei. And this shrimp it lacks eye stalks and black beady little eyes like a regular shrimp does have. Instead, it has taken its eyes and withdrawn them into the cephalothorax underneath a very thin, transparent carapace. They've lost the optics, so they can't form an image, but they can detect gradients of light. And we think that they are especially sensitive to very dim light. And we think that because the lack of optics means there's less material to absorb light, so the path from the stimulus to the receptor, from the source to the receptor, is as brief as possible, so few photons are lost by absorption. They also have a very high concentration of visual pigment, the rhodopsin. At least this is evidence based on shrimp from the mid-Atlantic ridge called Rimicaris exoculata. So a very high concentration of rhodopsin, so they have a lot of visual pigment, makes them more sensitive. And it's a very large organ. It covers the surface, a large part of the surface of the back, the cephalothorax of these shrimp. So those things combined suggest that it's very sensitive to dim light, which would make sense down here. The only light we think is down here under natural conditions is bioluminescence and light from the black smoker chimneys. Bioluminescence, to see bioluminescence, it's hard to imagine why you need such a highly modified eye. And so we think that they are probably using their photoreceptors to avoid being cooked by black smoker. So it's basically a remote thermal sensor. So instead of having to touch the hot water to know that it's hot, they can detect the glow from the hot water, and so stay safe. 44 shrimp. So in case you couldn't tell, this is like one of the most fun things a scientist can do. Come out on a research vessel. We're out here with a bunch of friends. We make new friends among the crew that we don't know, and other scientists who have come on board. We're a group of faculty, professors, research scientists, technicians, graduate students, often undergraduate students. It's like going to science summer camp. And even tasks like this that seem tedious are just a great pleasure to do. Best part of my job. The other thing that is really special about this kind of cruise is you bring along people with expertise in many, many different fields. And so you can be thinking about one thing and have a question. You can go to the person sitting next to you and ask them about it and get an answer from an expert. It leads to some interesting new ways of thinking about things, new experiments, new analyses that we can do to help us understand what's going on down here. So we kind of think together at meals and in the shipboard library where we all hang out up on the top bimini deck. Conversations are part of the important part of a science cruise. So Max and I tonight will be able to test the hypothesis that shrimp that have mouth parts covered by bacteria have bacteria in their guts, and shrimp without bacteria on their mouth parts have been eating meat. We'll have an answer for you tomorrow.
And Cindy will come up with the reason why they diversified into these two lifestyles. Which came first?
Yes.
The absence of bacteria or the easy prey of eating your own?
Yeah. So we'll in addition to looking at gut contents, we'll take tissues to figure out, make sure we're looking at the same species all the time, that we're not dealing with two species. I'm 99% confident they're all the same species, but we will watch. Yeah, look how clean the area is of shrimp.
Yeah.
To explain that.
Has anybody explained the Post-its?
It's not going to start here.
No.
Oh. Well chased down.
How many are we up to? 53. Closing in. Okay, so we are at, well, our count is 61 shrimp. You may have a different count, but we're going to stick with our 61. We're aiming for 64, so Chris Taylor is just about finished. I think the rate of shrimp collection went up. We're probably doing a couple a minute, several a minute.
You can get double now, Chris. That would be. Finish on
two
a
of
double.
them.
2 for 1.
Do you think you finish on a double?
Yeah, fine. It's fast. Good enough for their name.
Whoa, that guy slipped away. They've all gone into hiding. Well, they're not in hiding. They're all gone. So the very last two, we're going to have to shift our location just a hair, pick up the last couple.
Two to go. And I suggest that we can try to finish on a double.
4 now.
Yep, 62, and we had to shift because he got them all.
Sorry, Paul, who was it called? Thank you, mate. We should let the camera be on the matrix for the RV. NF1.
So we're just having a side conversation here about Chris's background. Apparently he did the breakfast show when he was at Cambridge as a student. That was as an undergraduate. So we can understand why he's such a good commentator for these dives. I know the audiences, we've gotten comments from people appreciating his live narration of events. Chris Taylor, the pilot, is wrapping up our shrimp sampling. The last two are recalcitrant. There goes one of them. One more. Oh, he disappeared.
Yes.
C'est fini. And one for good measure? No. Brilliant, Chris. Well done. Well done.
You don't need to finish on a
Yes, sir. Finish that sample 64.5 shrimp.
Done with the slurp?
Yes, we can stow the slurp, please. And I'll put you back over to Chris German. No, he's good. Yeah. All right. Thank you, everyone. I am slipping away.
Just for the record, at rate of productivity, that was 60 something shrimp in 40 minutes. At that rate of progress, he would have got his century if he kept going for an hour. Or he may have gone mad. Or both. So, we are now at 1:26 aboard ship. We are, I think, 5-6 hours into our dive. We arrived at 7:30 this morning. The dive's been going pretty well. We've been to 2 different sites, conducted some extensive sampling. I had a do list of 11 unreasonable demands from our pilots for the day. We have achieved 1, 2, 3, 4, 5, 6 of our tasks there. Actually, 5 of our tasks are completed. We're off to do the 6th one now. We're going to one of the first vent sites we found on this south side of Damm. We first observed it in 2011 on an Okeanos Explorer cruise, where Paul Tyler from Southampton, now retired, and I were the science leads actually out on the ship doing the kind of job we're doing here, and the watch leaders in the Nereus van, control room. But the rest of our science colleagues on that cruise were all based ashore at the Inner Space Centre. We know our work's being beamed ashore via them through the Exploration Now website, as well as from our own Schmidt Ocean Institute website. So, to the likes of Dwight Coleman and Catalina Martinez ashore in Rhode Island, a quick say hello to them. And also, of course, to everybody who was on the 1104 cruise. If you recognise Old Man Tree, that's where we're going to be heading to. We believe it is about 20 metres away, due west of us. So we're going to head over there. We're going to set up, collect some nice video, and then we'll be sending for Julie to come back in the van to do some more of her microbial sampling when we get there. John and Chris, when we get to the site we're going to, we'll recognise it because it has one of our markers there. And there's another marker that we think is a Shinkai marker, which ours is a yellow polypropylene rope with a big white diamond, like the X18 site yesterday. Or you probably didn't see that one. In fact, you guys probably saw the other marker yesterday. And then there's one that's like a tall white cylinder. Sounds good. So if we head over there, what I'd like us to do is we'll set up, get the cameras working, get some nice video, and then we'll go fetch Julie to come in and do the microbial sampling. So, full steam ahead. Ramming speed, and away we go. Beat the drums. What rate are we at? 53%. That's pretty good. We've gone on bottom 5 hours. We've done a long drive, done a lot of sampling. 278, I believe. So we've just been sampling around the Hot Cracks vent site. I think that was Hot Cracks number 2. I can see over on my right Hot Cracks number 1 disappearing out of view on a different field view camera, wider field of view. But where we're headed to now is a vent site that was named by Jill McDermott as Old Man Tree. We saw it but couldn't sample it with the Okeanos Explorer in 2011, and that just made for a really efficient dive program. And we came back in 2012 that because we'd done all the reconnaissance work with the Okeanos Explorer, courtesy of NOAA's Ocean Exploration Program, we came back with our jointly funded NSF and NASA crews in 2012 to conduct our first set of sampling, and now we're back here courtesy of the Schmidt Ocean Institute, not just revisiting places we already know about, but as you will have seen earlier in the dive this morning, discovering completely new vent sites, even in places we've been to several times before, or not exactly the same place, but within 100 meters or so. We're still making new discoveries, as well as being able to come back having had time to think about our first sets of results and conduct additional sampling and testing. So I think we're going to go off the edge of the hill if we carry on too far on that heading, so might need to bend a little to our right. I think we want to keep the topography in sight. I think probably what happens is from that rubble we go over a little patch of sediment anyway along the spine of the ridge, and then we should come into more boulders, but if we go too far, you can see on the left there, it just drops away into nothing. And one of the possibilities I'm beginning to wonder about now is this entire spine may be made up of hydrothermal precipitate. There's been flow along here, so it's not just the giant Von Damm cone that we're going to head up later on in the dive, but all of this spur that we drove along yesterday and we're driving along again today may be made up of it entirely from fluid flow along giant fissures penetrating deep into the earth, bringing the fluid up and precipitating chemicals to form the minerals that we see. The vent site is basically, I think, from what I remember, going in this direction. It's basically just over the top of the hill and then you drop down again.
Are we gonna have to come around and turn around, line up on it? Probably?
No, well, I don't know. I think we've approached it. I think it's kind of at the summit,
Okay.
at a local high, so I think you can approach it from multiple directions. We actually have some JSON video where we sampled it from one side and then we pivoted around and saw it from a different side as well.
Straight off on here. And it's at the top of this, you think, Chris?
I think so. I'm just checking the depth we have for that. For some reason we don't have that. That's the one depth we don't have written into our dive plan.
So our USB also showing us a ways away. Doppler says you're pretty close. I'm gonna keep going up then. Yeah. That would be
Why don't you stand by here? I'm just going to go check the depth for Old Man Tree and then we'll come back. There's a tube worm just on your right though. Let's go have a look at that. You can show that to people while we're waiting.
okay.
Yeah, if we're heading northwest, we're basically going the right kind of direction, I think. Is the pink dot Damm more reliable?
Pink dot is USBL. It just seems to be very stubborn. We haven't had to fix it in it, so we have to look at it.
Okay.
If I go in out of depth, it might come in.
Yeah.
I think the further complication is we're using the depths from Jason from last year, that 2373 depth. Can you look back in the log, Leighton, and see if you have a fix for when we passed over Old Man Tree yesterday? It would have been late in the dive. And if you maybe you could even search on, if Old Man Tree doesn't come up in the search, you could try marker, because we saw 2 different markers.
Big wall.
Okay, it was 2369 on Nereus depth.
Yeah, we're trying to do 70 depth now.
So there's a big wall there.
Yep, so I think we've probably gone a little bit too far.
And you go through that wall?
It's on the mound just south of that wall. So we may have to get on a reciprocal heading.
I got a Doppler position.
Yeah,
236, sorry, 2054, Y920. 2054 is 50 meters away.
yeah, that was where we left yesterday. What heading were we on? Does it? Oh, we don't have the heading there. So we're basically, everything is aligned on a northwest southeast line. So I suspect we probably came up west of Old Man Tree just there. And this lump we're just coming off of. So let's go back down here. I would circle around to the right, if anything, Chris, and come back down.
I'll like over my tents, so I'm gonna go down and then turn around.
Yeah, so do like a big loop around. But all our navigation says we're north of Old Man Tree here. And that kind of makes sense, because when we saw that sediment, we've gone over the west side of the shoulder.
So you want me to come around?
Yeah, what I would do is, if you came around, so you were going east to start with, if you come up to that, like, find the seafloor here, if we turn east till we're back on the seafloor at this kind of depth, then as we go across, can you see, like, the contours there where they would join up? If we pick up the rocky stuff and we head down the spine, that's what we were doing. That's when we first found it. Was so we 2-3. What? Can you remind me again, Leighton, what the depth was yesterday when Nereus depth?
What?
This-
Old Man Tree, USBL is at 2369. Doppler position is always at 2365.
No. So, yeah, we were at 2369, but we were flying. That was yesterday when we were on the run. So it's probably low 2370s. Hmm? Yeah. Right. So that's what we want to be, is the 2370 to 2375 is our target depth. Yeah. Why did we turn the lights out? Just so we don't want to be able to see? Or we can use these ones, right? It's, yeah, that's better. I can actually see things.
Port outer yellow.
Right, what's our heading now? So we want to come around more to east, please, Chris? And we want to end up on a heading of around 120.
Around what?
120? Maybe let's just follow up this hill since we're facing straight in and we've got these dead shells right in front of us. That's a good clue.
Yeah. 65 streams.
And this is pretty much, we're on the right range, right? Should be 20 meters ahead on this heading according to the... John?
Yeah.
Is that the right range and bearing to Old Man Tree from the vehicle?
Yeah.
Good.
Well, give them the Doppler position.
Yep.
It's not enough.
Certainly going into something.
There's something. We're on the right heading and there's something light ahead of us.
There's marking right
Yep, and the other one should be just the other side of it. That's not our marker, but let's get close to it.
there. Is this where we're going?
Yes,
Okay.
I believe so. Well, there were two markers. The
Diamond mark.
yeah, that's the diamond marker is actually right by the vent site. The other one, we're not sure whose it is or what date it is. It's hard to see what's going on because Casey's playing power management and flashing the lights on and off. I think he's turning into a disco on the seafloor.
You can turn on and off for me against my will.
If you
Yep.
do you're getting so much backscatter because the lights are right behind the channel. So do port brow and then port nose.
So, I think it's mostly because we're just coming back over an area we must have just missed before.
Yeah, he's also way out ahead.
Yeah.
Back
So we're coming up on the marker you see on the left is marker X19. Somewhere on the seafloor there, when we get up close, is one of these clear fluid sites, like the ones we were sampling yesterday called Twin Peaks. And then there's a second marker which shows other researchers have been here since us. We were involved in the discoveries here in 2011, the first time we imaged this site. Yep. We sampled it again in 2012. I think the active flow may be just in front of you to the left. And we're approaching from the right side because then we don't have the markers following us. Actually, I'm not exactly sure the other place it might be is up to the right. You can see the tall vertical marker?
Yes.
Just to the right of that is another up, so we should get close enough to actually look in our cameras to find out where the shimmering flow is, and once we get there...
Trying to scoot around a little bit.
Yeah,
So it's an album plate. Yeah, it's an album plate. It's actually a
it will be a Jason plate. Although it could have been... Alvin was due to come here in May, but that didn't happen. That was how I was meant to spend my 50th birthday. People said I should do something memorable. I didn't mind waiting an extra couple of weeks. It's still pretty cool to be here now. So we're looking for where the active fluid flow is. And we're looking for the active flow. I see it just to the right of the upper marker. See where the base of the upper marker is? Just to the right of that. Top right, just going off the top of the right. It's just gone into the darkness now.
It's behind this rock. you're saying.
It's actually coming out the...
Okay.
Where's my cardboard tube? There's something just there, and if you look right on the skyline,
Okay.
I'm pretty sure I can see Schlieren effect just there.
Yep. I see it.
We're not sure whose marker that is. I'm guessing it's probably a Shinkai one because it doesn't look like any of the UK markers. It's certainly not our one. That's ours on the left that we left behind. That could be a dive weight. We're not quite sure what that is on the seafloor. Hopefully it's not another one of Cindy's whale bones sitting right where we want to do an experiment. If we get up closer, we may be able to see writing. Yep, that's Old Man Tree. It does. So here we are arriving at the seafloor at an area we know as Old Man Tree. We were joking this morning that we got stuck in traffic and that's why our launch was 15 minutes late. Turned out it wasn't that big a joke that, in fact, while we've been doing our investigations here on the Mid-Cayman Rise at 2 different hydrothermal fields, our colleagues from Japan, or led by Ken Takai from Japan, but with international colleagues including some of our friends from the UK, led by John Copley, are also out at sea on the Yokosuka, which drove right by us this morning while we were launching. They are diving in the Shinkai submersible at the same 2 sites, so we're switching to and fro. We have 3 days at Von Damm while they were at the Picard site to start our studies. Then we switched sites for 3 days. Now we're back having 2 days at Von Damm while they're at Picard, and then we're going to switch back again in 2 days' time. Okay, so we've settled up. You can see it was a bit mucky here. We actually noticed that the markers that are here are actually blowing over in a pretty stiff breeze. In fact, you can see this Shinkai marker, which has been put right next to the vent site, so we had to work out how to set up, and it's kind of going to be blowing around in our field of view. But what we're going to do now is pick up our microbial fluid sampler, the super sampler, and get ready to do temperature sampling and fluid sampling here at this vent site. Actually, you know what? It's clearing pretty fast. By the time you pick up the super sampler, Chris, we'd probably be able to. We would then be able to. Yeah, probably. Could be a dead clam or something. If it's that bright white, it probably means it's a shell of something that's actually opened out and become disarticulated. Let's pick up the sampler. We'll get it in our hand and then we'll zoom in and then get some nice video of this site.
John, stand by my door.
So we're just getting ourselves ready to do our next little bit of sampling. So we're picking up a sampler. Cliff,
Yes.
if you're free, could you wander along to the wet lab? Tell Julie we're just moving the sampler out the way to do some nice video, and we'll be ready for her in about 5 minutes. So at the top of the hour, we'll be ready to do her microbial sampling. Thank you. So it's interesting that earlier today we were talking with Casey about how long it takes to get just a few, a minute or so of really nice video of different pristine natural environments. And you'll notice that the marker we left behind a year ago was placed about 1.5 meters away from the vent site rather than right on top of the system. Having a bit of trouble here that somebody else's marker is right beside the vent site. Maybe just let it go, Chris. Yep. No, let's just let that do what it wants to do. If it swung all the way around, we better see over the top of it. Okay, let's set up to do Julie's sampling, and if the thing blows out the way, we'll get some nice video afterwards. Or maybe we can reposition ourselves around that corner. So, time to do some microbial sampling. And to start that, we can probably... are you going to be able to see to actually get the temperature sensor in there properly? Well, let's do the best we can, and then we can actually check the temperatures and see how we do. Yeah, let's go fetch Julie then and get that started.
About what? 8. All right. We are setting up here at Old Man Tree, which used to look like a crooked tree, which is why we named it that in 2012. Looks like it's been run into.
24, 31, 38, 45, 47, 50, 56, 64, 71, 75, 82, 83,
And remember here we expect to be around 115.
94, 95, 96,
That marker you see pointing up toward the vehicle, we believe is a marker left behind by Shinkai 6500, which was
101,
here a few days ago.
102,
We've gotten 102 so far. Let's see if we can get
climbing, 103,
a little bit higher before we start taking any samples.
still climbing, slowing, but
Ken Takai is a microbiologist from Jamstack in Japan. He and his team, we've been trading sites back and forth during this expedition.
108,
So while one of us dives at Picard, the other one's at Von Damm, and vice versa.
110,
That looks good to me.
111,
I'd say we lock it in. We're at 111. Are you in a good place, Chris?
Everything looks stable right
Okay. Chris says everything looks stable right now, so we're going to lock it in. We're going to purge the pump. And hopefully get a sample. This is going to be Nereus 16. We're going to... we're going to start with Sterivex number 6, right?
Stairvex, we're on yeah, conversion, right?
So that'll be like a 30-minute or so sample.
Yes, correct. What's the temperature doing there, Sean?
Right now it is about 114 degrees.
Ah, perfect. 114 degrees. That's right, what we expect. Good job, Chris.
Very stable at 113.7.
Both Chris's. Good Chris, bad Chris.
Evil Chris.
Evil Chris. Sorry.
Evil Dictator Chris.
Evil
Supreme
Dictator
Evil.
Chris. And Pickles.
Stuff like-
Sugar Chris. Sugar coated Chris. Sugar boy.
Also known as the frosted flake.
So we are just flushing out the pump, and then we're going to be getting sampling here.
Sorry about that. Get ready to sample on 6.
Getting ready to start. Nereus sample 16 is starting at 1804.
Temperature is 111.
Temperature is 111. Max we've seen so far is 114 degrees C, and the reason I'm interested in this sample is because the upper temperature limit of life is 122 degrees Celsius, and so this is right on the edge of where most microbial life can live. And this is a good vent. It's a direct pipe to life beneath the surface, and we're just capturing it as it exits the seafloor. What happened to the Japanese marker?
It's right here, right beside.
Oh,
the-
right.
Blowing to and fro in front of us. They put it a bit closer than I do. We might step it back a little bit.
What?
We might nudge it back a little bit so it's not right by the vent.
What temperature are we at?
We still 114? Sure.
1112.
Perfect. You don't mean move the vehicle, you mean move the marker? What are you talking about, nudging?
Not right now.
Where have we had that before? I got written a news. The version of Jamming that you had?
What's our battery power at over there, Jonathan?
I bet you wish, is it? I bet you wish I was diving.
We are at 49% battery power, which is great because we've been on this. We launched at 6 a.m. and it's 2 o'clock. So we've been on the seafloor for 6 hours, in the water for 8 hours. We've only used about half of our battery power. So we are good to go for another couple hours before we have to make our way to the surface so we can do our vehicle recovery in the daylight. So the way we do the recovery is actually, I don't really know how you do the recovery when does the fiber get cut?
About 100 meters above the seafloor, we can do video transects up.
Okay.
We're looking at zooplankton living above the vent side, and then they cut the fiber pretty soon after that.
How do you cut the fiber? You just pull really hard?
No, there's a little cutter and then the actuator.
Up on top of the vehicle, there's a cutter?
In the flow pack.
Oh, so in the float pack that connects the depressor to fiber optic cable to another float pack with more cable and Nereus.
The flow pack is attached to Nereus
Okay.
with the armor, with a large cable.
Okay.
So the flow pack is just above Nereus.
Okay.
It's light fiber all the way
To the depressor, right.
to the
Okay. So Chris is telling me that the where the basically the fiber optic cable gets cut at the end of the dive, go up about 100 meters through the water column, and then cut the fiber. So we no longer have video or things like that from the vehicle, but we can still know where it is because we can talk to it with the transponder. And then so Nereus basically floats to the surface because it's positively buoyant at that time. We usually drop weights before we come up. And then they reel the depressor in with a winch. Nereus pops up, and we have a small boat meet Nereus, ties a line to it, tows it in closer to the Falkor, and then the Falkor positions itself near the vehicle. We throw out another line that the people in the small boat then tie to Nereus. So instead of being dragged by the small boat, it's now being dragged by the big boat. And then Nereus gets taken up into the A-frame back onto the ship. So it's a lot of careful coordination to get the vehicle back safely, which is why we are mainly doing it in the daylight. But Nereus does have flashing lights on it, right? It has a beacon so that you can do nighttime recoveries on it. And obviously, if we were ever to misplace it, it's bright yellow and it has lights on it, so it's easy to find in the big blue ocean. Easy being a relative term. So we're sitting here pumping a 110 degree fluid. 112 degrees Celsius, which is like 240 or so degrees Fahrenheit. Way too hot for you and me. But a good place to be a high temperature anaerobic microbe. These organisms are called thermophiles or hyperthermophiles. They have obligate growth temperatures above 70 degrees Celsius. And they don't like oxygen. It's not good for them. So they are likely early remnants of organisms that were here when our life first evolved on our planet. First first 2 billion years or so of life on our planet, there was no oxygen. Then there was the great oxygenation event around 2.8 billion years ago. But before that, all life on our planet was microbial. It was bacteria or archaea. And then with the rise of oxygen, only then did eukaryotic life, like you and me and plants and dinosaurs, become possible. So most of the evolutionary history of life on our Earth is actually microbial. The earliest evidence of life on our planet is at 3.8 billion years ago. Our planet is about 4.2 billion years. And while we'll never really know what happened way back then, it's fun to guess. And we can use places like Damm and other hydrothermal systems to do experiments and test hypotheses about how life might have originated and evolved, and what early life, geochemistry, rocks, and things like that might have looked like on our planet. So someone's asking me a question on Twitter. How many shrimp would you estimate per square meter in the vent versus periphery? Well, Erica, I'm not sure that's a question I could answer, but I do know that last year we did surveys with Paul Tyler from the UK, where we did very careful video surveys where the goal was to test, to count the shrimp. We actually did that. I'm now remembering that. I usually left the room when we did that. So I don't know the answer. When I see Cindy again, I'll ask her.
I think the sparse shrimp we were looking at just now is probably 5 or 10 per square meter. We're having to move to get more.
So that maybe
There were a lot around the cracks.
But in the cracks, it must be in the hundreds because there's several layers deep as well.
So we're going to say that sparse shrimp 5 to 10 per square meter. That seems reasonable to me. And then bazillions in the fence. But obviously getting an idea of exactly how much biomass these ecosystems can host is one of the goals of the biologists working on board. If you were to take Nereus and drive east from New York off the margin down into the abyssal plains, you would see very, very little on the seafloor except for sediment and more sediment and more sediment. And while there is microbial life and some eukaryotic life in those sediments, there's not a lot of organic carbon raining down. And so you don't see large congregations of vertebrate life or even invertebrate life like what we're seeing here at the hydrothermal vents. So these vents are really hot, oases of life. And they represent a part of the seafloor where we have new production of carbon. And that's why we see all these unique animals and fauna congregating around them. The rate of discovery of new species described at hydrothermal vents is really exceptional. Every time we find a new vent, we seem to be finding new organisms. And piecing together the puzzle biogeography of those animals is really interesting. I don't do that, but I think it's interesting. What's our temperature look like, Sean? Or Chip? 112. Sean, you don't have to stay if you don't want. We could just keep the same log running for all our samples. If we run into a problem, we know where you are. Okay. Sean is busy prepping the next pair of isobaric gas tights that are going to go down on our next dive. Our next dive is going to be at Picard, the world's deepest hydrothermal vents, just shy of 4,000 meters. So we're just
Longer on your sample?
Chip, what's our flow rate looking like?
It's like about 6 milliliters a second.
Okay, 6 milliliters a second.
7 milliliters.
So that works out to about 360 mils per minute. And we've been pumping for 12 minutes. Yep. So what I'm thinking is maybe I'll cut So we have a pretty rock solid, what does that temperature over there say? 112 degree fluid that we're sampling here. What you're looking at is the arm with an intake down into the vent, and then the marker that's sort of floating in our field of view was left by Shinkai 6500 probably just a few days ago. While we were diving at Picard, they were over here, and now they're back at Picard. So this one will be done in about 5 minutes, and then we'd like to do about 10 more minutes of sampling, and then we can be done. So in the control van with us, we've got Chris Taylor piloting, Jonathan Howland working on navigation and vehicle help. We've got Chip Breyer in the back of the room helping run the sampler. Fearless leader, Chris German's next to me, watching over the dive, making sure I don't do anything wrong, take anything illegal, leave anything behind. We try to be careful down here and not muck it up too much. And actually, there are now parts of the deep sea where, in Canadian waters, for example, there's a hydrothermal vent field site where you're not allowed to take any rocks or organisms, and you're not allowed to leave anything on the seafloor. You're only allowed to take fluids. And that's in an effort to have some sites that are preserved in their natural state where we can go and observe those changes, but not take anything away. And we're always very careful when we take large animals like tube worms and shrimp to only take what we need to complete the experiments. It's always strange when you're at one of these places and you randomly find a piece of garbage on the seafloor that was probably tossed aboard, tossed off some ship passing through, or something that fell off a freighter or something like that. We saw Reese's peanut butter cup wrappers a couple times last year. Sometimes you see barrels and tires and all sorts of strange things that you wouldn't expect at a place like this, and a reminder that the human impact on the ocean is far-reaching and wide. No corner is really protected from us. So let's stop that at 1824, Chip, and then we'll switch. Oh, I meant to stop it in 2 minutes, but that's okay. Okay, let's just do our last 2 minutes.
So Julie, after you stop it, I want to, before you start your next sample.
Okay. Okay. So we're concentrating the fluids here to collect all the microbes that are living in the fluid, collecting upwards of 2 to 3 billion cells. Microbes have a really large effective population size. They're clonal organisms. They grow by binary fission. Basically means the cell divides in two, and then divides in two, and divides in two, so they can have exponential growth when they're happy. And cultures in the lab can get up to 1 billion cells in a single milliliter. of water, which is pretty remarkable when you think about it. There's some lab rats of microbiology, probably heard of organisms like E. coli and things like that. And those are organisms that we use in the laboratory for molecular biology and other things like that. And also just to understand basic cell function. So we're going to stop that sample at 1824. We had a pretty rock solid 112. So they're going to... That was a Stervex. Yeah. Once they're done with... What's going on with the temperature when that jerk happened? Okay.
It's not all coming out of this side.
Okay, so we're switching to bottle number 7. Switching to bottle number 7. Let us know when you're starting that. We're still at 110, right? Okay, yep. Bottle number 7. Nereus sample number 17, starting at 1825.
Nereus sample 17.
Yep. So now we are filling a water bottle from this site with this 111 degree fluid. Old Man Tree. And it's right near marker X15, right? Is that what our marker was? Oh, X16. Japanese marker 5, but our marker is X16. Oh, Shinkai 54. Okay. We don't really care about Shinkai's marker. Temperature holding pretty steady at 111 degrees C. Hey, Chip, what's the minimum amount of time that we can fill this bottle? Because I want to fill 2. Can we do 5 minutes per model? Because that would meet the 10-minute. Okay.
So the time on board the RV Falcor now is... So the local time aboard the ship is 2:27. We are taking the last samples that Julie really wanted to get from the Von Damm hydrothermal field at this site, and it's just as well because we're having a technical problem with our arm. We worried about. The team worked through the night on 2 different problems with our manipulator. The thing about Nereus is we only have the one manipulator, so without that we are completely stuck in the water in terms of what we can actually do in terms of scientific sampling. So we're taking our samples as quickly as we can. Chip in the back of the room has turned up the pumping rate to make sure we get the 2 samples we need. And we'll probably be out of here in about the next 5 to 10 minutes. We were making really good progress with our dive. We've had an excellent dive so far. We're not going to get all of our objectives done, but we won't have had a bad dive when we come off the bottom in the next 5 to 10 minutes. Since we are here, is there any chance we can zoom in on the vent site, or do you need to be looking at the arm just now? Let's do that. Let's. It's not easy to get an unobstructed view, but maybe if we switch to our zoom camera, can we do that?
Yeah, I'm gonna secure this one.
Okay, stopping that sample at 1829. And
So we have.
so skip to bottle number 9.
Okay.
Going up. Over 10. Yep. Let me know when you start.
That's nice. Can we? Maybe we can focus in on some of the shrimp in the near field. Beautiful. Did you start sampling yet, Chip?
Not yet.
Okay.
Yeah, I'm gonna start sampling them now.
Okay, starting that sample at 1829.
So 1829.
So the temp's
This
going
is
to 104. Remember? Okay. I know,
a
but
whole.
what is he doing when he's pumping?
What is it? Right now we're pumping,
I got pumping high speed.
109.
and it's 109.
Fill up.
Great. So what is that pump rate? Like liter a minute?
This is, let's see, what's 25 times 3?
75?
75.
75 milliliters a second.
So that is
4 liters per minute?
Yeah.
4 liters a minute.
That's the maximum.
Okay.
So we're pumping this water through real fast to get this whole water sample. It really is a super sampler. It's turning into a superstar sampler now.
It's always been
So-
a superstar.
Since we're coming to the end of this dive, what I haven't done all day, but I'm going to do it now, is wish a happy birthday to my big sister Maddie. When I turned 50 earlier this year, she told me it was about time I got a real job sometime. But the bad news for her is we're sitting here in front of a vent site very aptly called Old Man Tree. I didn't know we were going to be coming here, but now I've become an old man, Maddie. I'm afraid I'm still not grown up, and I'm still not going to get a proper job because this is way too much fun. So I'm going to be sticking out here in the middle of the ocean for as long as I can till they drag me kicking and screaming off to some place where they make me wear the special T-shirts that Leighton keeps offering me. And but sadly today, it's like all dives have to come to an end. It's probably just as well things do go wrong every now and then, and we have to leave the seafloor because otherwise it's just because the sun's setting I get dragged kicking and screaming off the seafloor. If you've been reading my blog, you know that sometimes I do get overtired and I scream and shout and pout,
Okay, we're done.
and they make me go sit on the naughty deck.
107.
But in the meantime,
Okay.
we're getting ready to wrap up the science here. That last sample is taken.
Yep. Do you just know the pumps on this last
That was 1831. We got something of everything we wanted to get today from this vent site.
It was up to like 4 liters per minute.
So, Chris, we are happy to stow the arm as soon as you are ready to stow the arm.
I don't see it anymore.
Yeah, I'm not seeing much.
What does that mean?
Might be
So we're reaching the end of our time here at the Von Damm vent site. We will be finishing our dive shortly and rising up from the seafloor. And just wait one moment while Julie and Julie exit the building. So this has been a great place to actually end our last dive at the Von Damm site for this cruise, if that's how it proves to be. We're planning to go to the Picard site for the next 2 days. The cool thing about the Von Damm vent site is, in fact, this very vent, the Old Man Tree site, was actually named on our first experience of using telepresence to explore the deep ocean with the Okeanos Explorer in 2011. It was named based on a conversation I was having with our graduate student, Jill McDermott, when she was actually based in the University of Rhode Island's Inner Space Center. I was out here and we were discussing live over telepresence two-way audio what we thought the event most reminded us of. So that's how it got its name. And as you can see, it got discovered then. It's been returned to 2 or 3 times since. We came back here with the Jason ROV in 2012. The Shinkai has clearly been here earlier this week. We're here again today. And there's a very good chance that the ROV Jason's sister vehicle, Isis, was here back in February this year as well. We know many of these vent sites have been visited on multiple occasions with different vehicles. So we're just having some quiet time here above the Falcor with the various engineers, including Andy Bowen, the director of the Deep Submergence Facility, doing a bit of evaluation of exactly what's the problem is with the arm and how big the problem is. That'll evaluate whether we can carry on diving and searching around new sites, or whether we need to return to the surface straight away. We'll let you know as soon as we know. So, for those of you following along the dive at home, it is coming up for 2:45 aboard the RV Falcor. We're on US East Coast time. It is Thursday, June the 27th, 2013, and we are stationed right in front of the Old Man Tree event site, part of the Damm hydrothermal field on the Mid-Cayman Rise. Situated in 2,300 meters of water. We're actually at 2,370 meters right now. We've had a great dive so far. We've conducted a lot of sampling. We found some new vent sites we'd never been to earlier in the dive at the very start, around 6:30 this morning, which is, I suddenly realize, already about 8 hours ago. We collected new fluids. We collected microbial samples from those northern sites. One was called shrimp buttery. I've already forgotten the other one. It's been a long morning. We then transited around to the east side of the Damm spire and conducted some more sampling, both microbial sampling at an area called Hot Cracks. Then right after lunch, we then collected, I think it was 65 shrimp altogether from that area for a range of different scientists, including some who've actually only joined our science team since the cruise started. And we've done some further fluid sampling here at the Old Man Tree site.
Are you ready?
And we've now been consulting with our engineering team. It seems like, although we have a problem with our manipulator, we shouldn't use it anymore today. We don't want to put any more wear and tear on it. It seems like we are going to have permission to carry on driving around and actually carrying out some exploration on the seafloor. And we'll keep a track on how the instrumentation is doing. Okay, so the good news is we get a stay of execution. We actually get to continue this dive a little longer. And before we leave, we're just curious to find out what that little spherical object is behind the marker in the far distance. If we look up, it's what we want to look at. That's what we're zooming in on.
No, I think it's
one of those little orbs.
It's a little Japanese version. That's it. That's our zoom camera, and that's it in focus. It might be attached to, because what's this black thing just below that? So we are completed with our sampling at Old Man Tree. We are now going to see if we can find sites with live mussels. Although we can't use our arm to collect any more mussels today, what's kind of convenient is Cindy Vandover already has her flights booked to come back out to the Cayman Rise and continue her research here later in the summer with the Ocean Exploration Trust aboard the exploration vessel Nautilus. So they'll be coming back with their ROV. Tomorrow and the next day we're going to be having our next 2 dives at the Pickard hydrothermal field. But what we're going to go and do is see whether we can't find live mussels at some locations that where we've seen photographs from Nereus from leg 1 when it was in AUV mode that apparently revealed the presence of mussels on the seafloor. So the target we're heading to, John Howland, the target we're heading to now is Ravelin number 1. So if you could give Chris a range and bearing, and that's where we're going to head.
Chris? Yes. No, I would know Chris from
So we have 44% battery level still in the vehicle. We have about an hour and a half until we have to leave the seafloor. And we have interesting things to go look for. And we're going to be setting off as soon as our pilot and co-pilot are ready.
Miles going to head off the van.
We got a plan. Chris and Chris in the van. Leighton's left us, John's left us, Cindy's still here looking nervous. She trusts us. If we do find live mussels, you have to bring one back for my sister for her birthday because I didn't pick her up a present yet. And we can't with the manipulator. Cindy rocks. I hope my sister didn't, then she'll know that I was even too cheap to get her my own birthday present, that I stole it off my friend. But she's very long-suffering. She actually has some sediment core that I collected from the Rainbow Vent site when it was her birthday about 15 years back. So I think even the labeling's run out, but she still knows where it came from, so that's it. We'll give her a rock from the Cayman Rise to go with the mud from... See, it's going up in pressure, right? If we just go deeper and deeper, one day maybe I'll be buying her diamonds, but... just some unconsolidated hydrothermal rock will be pretty good for a starter. It'll have a bit more shape to it than a push core. Okay, Chris, we are headed northeast, and my best estimate is it's going to be 10, 20, 30 meters to get to the Ravelin site. And our target depth when we get there is 2,383 meters. In fact, there are 2 sites.
So we're going to be going down. Okay.
Yep, I think we're probably going to go. Actually, I guess we're over. We probably were right on the spine of the ridge. We should probably, if we go out, instead of going straight there, if we actually went maybe more east-southeast, sorry, east-northeast to start with, and then we could approach it from the south, then we'd be going uphill again when we get there.
So if I turn my head east-northeast,
Yeah, if you made it like 070 maybe. If you went 070 for about 30 meters, and then we could probably turn due north.
so we'll probably come down then and then.
Yeah. And in fact, what you could do is, if we look, you can see some sediment straight ahead. If we aim for that, there should be a sediment rock boulder, and then we could turn east along that, and then we'll be flying over soft sediment, and that'll be nice sediment to fly over. Then we won't worry about bumping. Then we do a little off-piste, just ski down. Bend the knees, jump to the left, jump to the right. Make this little robot dance. So we're a ways away from where I think the exciting site's going to be, Cindy. I think these are the dead mussels we flew over yesterday, but you got a good view there and keep your eyes peeled.
Do you want to look more down or more out ahead?
I suspect Cindy would like to look right down, so that she do see animals, and I'm going to find my way by braille. Oh yeah, actually let's pan up a little bit, because the view of the basket is a wasted view. There we go, stop there.
Like that?
And we know the hose at the front of the basket's only just out of our field of view. So if we turn right here, Chris. Yeah, those are the ones we drove up over yesterday. So that was our best hits, and now we're going over to 2 other locations in our nearest survey we found. There were 3, it turned out there was the photographs you saw, the mussels in, plotted up in 3 different geographic locations, so we went to our favorite place first. But here are 2 other targets we have.
I knew it was something that you were actually not end up underneath a desk or a table at some point.
Yep. John Howland is climbing onto the table, and it's making the basket shake. Yeah. I did, and he had that idea that we'd stop every 10 meters and do a little square wave dance. Well, I'm trying to think what you can do with a tether, because we mustn't go back on our tether. I think, well, spiraling up into our cable probably isn't a good thing. We might have to do quite a long... Yeah. We actually we come up at 25 meters a minute, so the speed he wanted to come up was exactly what our speed is. Not easily, because we drop the weights and come up with... Yeah, so unless we came up... Let's see how much energy we want to spend trying to find the mussels for because those are all those things are things with an ROV with infinite power can do much more easily than a battery-powered version. Yeah. Yep, that'd be the bison of the deep. When we came here a few years ago, we actually went and dived on another cone-shaped system about 1 kilometer away from here that was completely dead, but Paul Tyler and I then decided we wanted to hightail it from one site to the next. It was our last dive, and we actually found a new vent site during the course of that, but we spent about an hour driving over barren sediment. We didn't get to see much at all. We were talking about somebody in Wyoming. Oh, actually, it must have been Mike and Bobby from Wyoming were discussing how it looked as bleak as the high prairie. And then we actually found a cluster of about 50 holothurians all lined up with the current, all roaming together across the seafloor. So we just figured it just got even more like Wyoming now. It became like aerial photography. Ravelin 1, please, yes. So yeah, if we continue out due east, if we get ourselves to where our depth plus altitude gets to 2390, then we could take a sharp left, or bank around to the left and come back in on a heading going something like 330. It's our favourite heading in this site, right? Different day, same heading, 330.
330. Yeah, so this goes up.
Yeah, we should be like, I think we're in like a gully. Can you see how the contours on the DVL come in and out?
Looks like a gully over here.
Yep.
So should I, is that the direction you prefer?
Well, yeah, because we're not deep enough yet.
No, 2384. Yeah,
Right.
so-
So we want to get to 2390 would be my ideal. We'd fly out. Yeah, so now you can see Ravelin 2. So in fact, I hadn't seen that earlier. So if we're at the USBL point, we go a little bit. If we go a little bit further east, then we can turn around and line ourselves up. So we're going to go through Ravelin 2 and Ravelin 1. If you project back, John, like a vector that went from Ravelin 1 to Ravelin 2 to down to the southeast. You see what I'm saying? Let me come over and point. So we've headed out to the southeast corner. Can we drop down on that? See some light discoloration. There's some shells in there. Let's go have a look at them. How good are my eyes, huh? Hmm?
Well, it's incredibly different, but not so bad.
Shape or color?
Colour.
Yeah.
Black.
Yep.
Do we want to zoom in on these?
Yeah, sure. So what we've done is we have 3 different locations where we picked out where we saw photographs when Nereus was flying to and fro in AUV mode on this corner of the mound. And we just noticed they all basically are in perfect alignment once we actually took the photographs and asked where the photographs were taken from. Yeah. And Cindy's busy examining these mussel shells, looking for evidence of live ones. We're going to be connecting the dots going through these sites, keeping our eyes peeled to the seafloor. And I think that's good, Chris. Thank you for the zoom in.
That is zoom. That's
Yep.
the zoom camera.
Quite. So I think we can go back to the wide angle. And what we're probably going to want to do as we get lined up now is basically follow this heading. So we'll connect the dots from Ravelin 2 to Ravelin 1 to the best bet mussels, which is just in the top left corner of the screen.
So I'll bring my head around the last one
Whatever that-
lines up.
Yeah, if John can give you the vector that would take you through those. And what we'll just go do is just proceed slowly uphill. And what we'll do is we'll sing out every time we see something interesting on the seafloor. We'll stop you, if that's okay, and we'll
That's fine.
go down and check things out. Looks like there's a little light patch. Is that kind of in the right heading just there? It's probably a bit further. It's not quite the right heading, is it? Okay.
See how there's a fish up there?
White one in the top.
No, there's
Oh, let's go have a look at that.
something right in that crack.
Mhm. Anytime we see anything interesting biologically, it's worth checking out. And a Galathiid is not a crab.
Okay, we're on the right heading, Chris?
Okay. It's snowing on the Cayman Rise today.
Sorry about that.
No, that's.
There's this-
We'll be back through here pretty soon and back into clear water. It's the trouble with me making you do those handbrake turns, right? So we're heading forward, proceeding uphill. We're about 2 meters off the bottom at the moment, in a water depth of 2,384 meters. We're heading towards some sites where we have seen fluid flow on the seafloor before. We've seen photographs of mussel shells, and what we've not been sure about is whether there are actually any live mussels in these locations. So that's what we're hoping to find. Do you want to go a little bit north to get out of this, and then, because we don't want to go too far without being able to see the seafloor. I see the seabed ahead. We're currently at an altitude around 3 meters. We're going to come through the murk and back down to the seafloor, and then we'll turn back onto our northwest heading.
Turn off that starboard bow. So this is the problem with having a vehicle that's too heavy. Is when you have to thrust down, thrust up,
Mhm.
it starts that way.
Once we get past the sediment and get back onto the rocky stuff as we come up the slope, it'll improve.
Let's get safe for a second.
Mhm. So the time now is 12 minutes past 3 on the RV Falcor. We are in the same time zone as the east coast of the United States. We've had some great dive time on 3 different hydrothermal fields so far today at different points around the Von Damm site. We're now on our way to 2 more, and then we're going to head up to the top of the spire for the end of our dive. We are currently just gone off the edge of the mound onto thick layers of soft sediment. We're away from the hydrothermal deposits, and that's what you see at the moment. Is we've stirred up a little cloud of our own making. So it's a matter of getting out of that, and then we'll be continue on our explorations. So we were trying to wait for the sediment to clear out of our way, so we could carry on the heading we were on. And it's not really happening. There's enough light, fluffy sediment beneath us, and our thrusters are pushing down. It's just continued to stir it up. So rather than drive straight forward in fog and crash into the hillside, we're going to turn to our north along the contour. And we know that there are tongues of rock and rubble that come down off the hillside. We're basically on the east side of the Von Damm Mound. So we're going to turn, head due north until we come into clear water and we can see the seafloor again. And then once we do that, then we'll turn back uphill and make sure we don't get too close to the seafloor until we run out of this soft sediment and get back onto the hydrothermal deposits that we're interested in. And you can see there's rocky seafloor and that not that far ahead. So let's aim for that at constant depth. Do we have a good USBL position at the moment, John? Is that where you have the cursor just now? And is it at the pink cursor rather than... Or would you believe the white icon of Nereus? So we're pretty much right coming up on it right now. So let's look up high. Okay. Okay. Well, now we're over rocky stuff. Can we get a little lower, closer to the seafloor? Because even if we ride out Ravel in one, well, I think two things to bear in mind. The kind of fluid flow sites we see, you can sneak right up on them and not even notice they're there because they're pretty subtle, because they don't generate black smoke. But the flip side of that is that if we'd had light mussels when we were there sampling fluids for Julie last year, we would have seen them. So the mussels and Ravelin 2 and Ravelin 1 aren't necessarily exactly coincident, they're just close by. So if we happen to actually drive right over a vent site and recognize it, it's a happy bonus and we'll do some photography, but if we don't, we'll just keep going because our main thing is to look for the mussels. I think the truth is the hillside is peppered with these hydrothermal fluid flow sites. As we found this morning, we actually found new sites we'd never seen before, even though we'd been 5 meters away. So we're now at a depth of 2,381 meters, working our way northwest up the east flank of the Von Damm hydrothermal mound. We're at an altitude of 4 meters, and we're on the search for live mussels. See a Galatheid crab off to the right. Another Galatheid crab coming up on our path. Sorry, I keep saying Galathead crab. It's Galathead squat lobster. 20 years of saying the wrong thing. It's a hard habit to kick. Do we see any of the... Oh, there's something interesting. Let's go see a vent site. That's probably to within 5 meters. Could be Ravelin 2. Our depth is 2380. I saw some shrimp there. You think it's more like Ravelin 1 than Ravelin 2? Let's call it Ravelin 1.5. If we get up close, I'll see if I recognize it. There was one site like this that Paul Tyler and I found right at the end of it. Oh, wow. Actually, that's a pretty big site, huh? For the number of shrimp there, that's actually... I think this was probably a new site, different from the ones I've seen before. Actually, you know what? It's very sad of me, but I think I recognize that stump of rock right in the middle of the field of view, like the next ridge over. So I think we can, if we could drop a marker here, please, John. Let's call it Ravelin 1 question mark. And what we have here aboard ship is we have all the high definition video from when we were here previously and we collected samples. And yeah, I think there's a little chimney right in the foreground just here. So let's zoom in on that. And we might have to call Casey in from the van to tell us the temperature. Because we can't use the arm to measure the temperature, right, with the temperature probe, so... But Casey's been pretty good at eyeballing temperatures for us so far. So those are the little gastropod snet. Is that there? Looks like. I think, yeah, just that, yeah. I think just disappearing off the top of that. Seems like the field of view is pretty sharply focused, right? Oh, in fact, there's two chimneys. Can you see there's one at the base of the... in the middle, and little stump there, and then there's the second one higher up. So in fact, Jill probably knows... In fact, okay, now I see three. On the bottom of the field of view, I see two little sawn-off stumps. One right in the center, one a little bit to the right. And then there's a third one in between those two further up. It's like the apex of a triangle. There's one of the sites I recognized when we were flying over it with Jason. I think this is one of them we actually did come back to and sample, but I recognized the big yellow stump of rock that was right nearby. And see, Cindy just names all her shrimp when she collects them individually. And I get... It's my theory. And Julie's trying to come up with 1 billion different names for all her microbes. And I guess I'm enough of a geologist at heart that I see individual lumps of rock and recognize them. Oh, there's some of those pink shrimp there as well. Can we zoom down and look at those? And I'm going to give the microphone to Cindy to tell us something about the different species that are here. Would you like to do that, Cindy?
Okay, so in the field of view, we have two shrimp that are in the genus Lebius. So do you see the ones right middle screen? And to the right, there's 3 shrimp now I see walking around. So they have that kind of greenish, yellowish, whitish patch on their back is their gonad. So these guys have green gonads. It's also looks like their hepatopancreas might be the red part. These are shrimp. In this case, these do have eyes. They have eye stalks and black beady little eyes. We imagine they are predators, building the case for for that, or omnivores is probably a better way to put it. They'll eat whatever they can find, scavengers. And they are so a different genus, different family even from the other shrimp at the vents. They tend to be found in cooler waters, more on the periphery, relative to the Remicaris hybaci. So the Remicaris hybaci you see at the top of the screen. If your eye is getting good at picking these guys out, I can now count 4 Lebius in the picture, Lebius shrimp. Of course, there's a little eel pout watching out there. We think the eel pouts, well, they probably eat maybe shrimp, maybe snails. We've seen places where the shrimp are sitting surrounded by swarms of shrimp with a little perimeter of shrimp barren area, which suggests the shrimp don't like to get too close to the eel pouts. Very nice spot. Nice little park there.
Fluids again, please. And let's just hold that. Let's get the chimney. Actually, yeah. Beautifully positioned, Chris. No hands, Chris. No hands, Taylor. Setting up the photo shoot again. So, could we just pan up a little bit? I'd like to have the base of the 2 little chimneys. Just if I could have those down at the bottom of the picture, so we can see the fluid rising out. Stop there. And let's give that a sample number, because that's a pretty bit of video. Actually, to set my shoot up really beautifully, could I go a little bit to the left as well? Because you can see the plume is dispersing over to the left. So we pan the view that way. There we go. Super. Yep. And let's hold that for about a minute. Yep. Yeah. Cliff spent about 10 minutes this morning so we could actually get control of the iris and the focus even when we're in the zoom. So it's really come together now, so taking the opportunity to collect some pretty pictures when we can. Maybe that's it. I can't send my sister a present for her birthday, I can send her a video instead. I'll find a way to compress this. She knows I'm a very, very sad man, and she knows. She used to have to take me to school when I was 6, kicking and screaming, and so... One on one. That's how you count anemones. Anem1, anem1, anem1 on 1. Many anemones. Can you say that? I think Cindy's almost had enough of my commentary for the day now. Anem2, anem3, anem4, anemone. Oh, see, I can't say it right. I get past anem4 and I get that anemone. Okay, well I managed to keep Cindy laughing for the full 60 seconds of the highlight video now, so thank you, Chris. Let's rotate back to wide field of view. Yes, please, Leighton. I'm just going to get our XY coordinates while we're here. Could somebody shout out? We are 2058 in X. 942. There's shrimp all over. There's all kinds of stuff here. It's a very, very pretty sight. And right, onward and upward. Let's go find more cool stuff. Just checking out the arm, make sure everything's still okay there. And assuming all is good and we can carry on exploring, we'll carry on up the hill. We still okay to keep going, Chris? Okay, then let's camera up a little bit. And I guess the correct heading that we want to go is straight ahead over all that fluid flow. So maybe we want to scout around a little bit to the left. don't want to actually drive right through the thing and melt a little optical cable, right?
So if this is Rattler 1,
Yeah, if we're at 2380 in depth, I think there's about a 5 meter offset in our recorded depths. Well, that's hard to call. 2048, actually we are... Very hard to tell, but let's go on this heading and see what we find. We either will or will not find another vent site like this about 10 meters uphill from here. Cindy's just frightening Leighton on my right again, reminding everyone about how scary that big fish was. I'm going to tease Cindy now and tell her I was telling people this morning I stuck my head in the jaws of that fish when it came up, but it was actually my little finger only just managed to fit in there. What are these things on the seafloor right here? Like those white things? Those look like animal shells, yeah? Wouldn't it be nice if they were mussels right here? So if we nudge up to them and then go to zoom again, Cindy, would you like to do that? When we're getting close enough, if we can zoom on those. Once Chris is ready, yeah. And it looks like there's some more. Yeah, should we do the lower patch first, and then there's some just straight off the top of the screen as well. Oh, it's clearing. Is that a clamshell, Cindy? That pale white one? What are those? Impossible to tell. Okay. Mussel, okay. Don't look very healthy, do they? Can we begin to pan up, Chris, and see if we can...
just up above this.
Yeah. Oh. Yeah. Just rock. Do you want to go to the wide view? Then we can work out how to recenter. They were kind of sort of straight up from the first set, weren't they? At least in the wide angle field of view. Not that far up. No. That was them up there, right? Left of center. Yeah, I think we looked at one lot down there, so it's maybe we did see them all already. Okay, let's carry on onward and upward. And there's been a lot of mussels living here. Some over the.
Ready?
Yep. Do we have a time that we have to leave the bottom to be on the surface for 6?
I don't know. How long did it take us to ascend yesterday? James is James? Oh, James is
Casey, do we know what time we have to leave the bottom to be on surface for 6?
By 6, so I would say that in an hour and 45 minutes, probably.
Okay. And we've got 40 minutes, and we have plenty of battery, I believe, on 40%. See if we keep ourselves moving to make sure we get to the top of the spire and get the chance to do. So we're going to try and see whether we can't pass through that Ravelin 1 waypoint, right?
Do you want me to turn?
Yeah.
Where my caliper is?
Sure. I suppose if we take the USBL fix, we're already past that, right?
Yeah, so one hand will go wrong, the other one.
Well, let's not go downhill, so like
Well, right.
some more white coloration down there. Yeah, it's things being shed from the vent side uphill that's then spread out over the sediment down that's accumulating in between times. Yeah, there's shell fragments as well as rock fragments, aren't there? I think the stuff that's lighting up white is probably the translucent interior of the shell. That makes sense? Or like a pearly inside to it. I wasn't on that cruise. I think it could be a large shell coming into view on the top. But we're getting more of these galatheids. Maybe we'll get lucky and find a tube worm field on the way up the hill. On a galatheid? Yeah. Want to go have a look at that? Go get a pretty picture? Yeah. And the crab hanging on the underside of the rock. Guess you got a choice of two, right?
We'll see if I land.
He's shy. Is that going to be easy to get to if it's on an... Oh,
Yeah, it just takes me a second to figure out what my bottom is.
okay. I think we'll get the vehicle set up and we'll work out which one's going to be the easy one to look at. Depends which one we can shine the lights on, right? Dial down the iris. So, Mr. Galathead, what do you have to say to the watching world? Turn out the lights. Get my good side. Yeah, it really is a shy and retiring Galathead, huh? Settling out nicely, isn't it? Let's give it another 30 seconds. Or so. And then I think our fixes would put us, no matter how we tried, we're uphill from that ravel in one sight. So I think we'll probably just hightail it up the hill and get up to the spire, and then we'll do another one of those video surveys off the top of that to end the dive, if that's okay.
Same place we were yesterday?
Yeah, you know, we've got the camera dialed in so nicely. Let's go back up there. Go back up to that spire, get some nice video around the orifice to the vent. And then we could do some... no, that's fine, thanks.
Did you guys see the shrimp?
Scary shrimp. Oh, yeah. Shrimp in silhouette.
So go up there.
Actually, yeah. Why don't you give that a sample number, Leighton, as a bit of a highlight. Yep. So yeah, Chris, from here, why don't you make your fastest and most comfortable route to get to the top of the spire? And if that means coming off the bottom and flying relatively high up.
If I had Casey drive for you,
Absolutely.
I just got an email from Daniel having some trouble with the battery chargers, and
Okay.
I'd like to go address that.
Absolutely. Hmm? 4:15, and we'll be on the surface at 6. And, oh, John, could you call the bridge back and tell them we're no CTD tonight? Hmm? Oh. He's got to go do some stuff with battery chargers.
Best haste to the top of another spire. Takes me a little bit of transition.
So we're
Yeah, we've got the camera. After Cliff did all that work with the cameras, they're dialed in really nicely. So if this is our last dive, we want to go get some really nice video from that area. If you're up for that. So, whatever your best speed is to the top of the hill, number one. Good Chris has left the building. He's probably gone to listen to Elvis before Cindy can. He's gone to check out on our battery chargers. So Casey Machado has taken back over the pilot seat for the last 30 minutes of the dive before we head up to the surface. So we're about to go and make best speed northwest to the top of the spire. And so we're going to end the dive by going to the central spire. We haven't seen that all day today, but we have our cameras dialed in. We've stowed our manipulator arm ready for some repairs it's going to need when we get back on deck this evening. But the rest of the vehicle is in great shape. Videos working, cameras are working the best they've worked at this site, and we have some pretty spectacular stuff to go look at to end the dive. So that's exactly what we're going to go do. Yeah. Just in front of us.
Switch to the wide angle.
Yes, we can.
Oh, okay. Casey, can we, before we go anywhere, sorry about that, just on the bottom left-hand corner of the screen, if we could check those out. Cindy's already scoping out her future sampling sites for her next cruise. So that yellow-brown coloration is the good indication of... Oh, sorry. Okay. So Cindy just with a keen eye spotted some yellow coloration in some of the shells here. Wonder whether some of them might be alive, so we're just having the closest look we can. Can we pan left at all? Yeah. Would you like to drop a marker here, Cindy, so we've got a chance to navigate you back? John, could we drop a target in the DVL nav here, please? This is a... Cindy's going to be back here with... No, it's just... Well, it's... Oh, okay. I mean, it's going in as a marker. Yeah, so... Sample 21. Okay, did you get that, Nathan? Leighton, sorry. DVL, it's called DVL target number 13. Yep. Happy? Great. This kind of makes sense, Cindy, because we're very close to those Ravelin 1 and 2 sites, and they were picked out as being near to where you'd seen photographs from Nereus from the AUV dive. Yeah. You good? Okay, thank you, Casey. Let's get up to the top of the hill. Do-do-do-do. And what I'd like us to do when we get to that hill would be to come in around the east side and approach it from the north. Because then we'll have the big orifice will be right in front of us. Yeah. No, up round to the right. So if you came like, you see where that arrow loop marker is? Target 6? If we went through there and went north through there, then we could spin round to our left and come out. Does that make sense?
Can I ask you something real quick?
Sure. Can we afford any more light while we're coming up? How's that? A bit better? I think we're flying slightly higher altitude now, that's why it's hard to see so much. Yeah, exactly. Well, we want everything because we're scientists, but yeah, top is more important within the time available at this point. Big shells just there, look. So we are headed to the top of the spire, which is actually deceptively further away than I realized. It's 145 meters. It's probably something like a 10-minute drive, do you think? And we are climbing up from 2350 meters to the top of the hill, which is at about 2300. So we're going 150 meters forward and 50 meters straight up, about a 1 in 3 slope. But it's probably only going to get steeper as we get closer as well. Leighton, could we switch to the navigation screen for a moment? I think so, because I think for people that are watching on the internet, it's going to be dark and gloomy picture to do the expedient thing because we have a steep slope coming up. We'll just show people where we're headed and what's going on, and then when we get closer to the seafloor, so you can see there, Nereus is down in the bottom center of the picture. We're heading in a northwest direction. We're heading towards, the way we're headed, you can see there can be steep contours closing on us. Pretty soon, we're heading to the target 6 is called Arrow Loop, which is a area of fluid flow on the east side of the summit. And then once we get there, we're going to spin round to approach that central little red contour from the north side, because that's where the large orifice is of the vent site. So we're going to go there, do a little bit of filming right there with our cameras dialed in, and then we're going to end the dive with a zooplankton transect up above the center of the vent field again. We had a first go at that yesterday. We had some good ideas from Brendan Phillips, has sent them out to us, and we'll try and implement some variations on that theme tonight, see if we can do a even better job than we did yesterday. So, switching back from the navigation feed, we are back looking at the seafloor. Casey had us rise up to gain some altitude and then drive forward. We're now getting to where it steepens up even more as we move forward. Plenty of boulders in sight here, not much sign of biological activity this far down the central cone.
So we want to approach from the east going
No, we want to approach from the north going south. Yeah, I think if you made a beeline from here to that point 6 that's called Arrow Loop, it's kind of northwest, that would actually bring you up around the east side of the cone. I think the last few days we've come up so far over to the west, we've ended up coming round and coming at it from that side. But it's basically when you're facing into that big orifice at the top of the chimney, your heading's about 180 pretty much. But since we've done all our work this side, I don't think there's much point taking the tether around the far side and tying a knot round the spire. Yeah. Really is a huge edifice, isn't it? I think they're either 5 or 10 meter contours. We can count if we're at 2350 now, and it's 2300 at the top. So there's 1, 2, 3, 4. They may actually be 10 meter contours. And those boxes are 100 meters, so we're going to go up 10, 20. Yeah, we're going to go up 40 meters in the next 50 meters forwards. Yeah, that makes sense. It's about a 45-degree slope. Yeah. And the current thinking, or one lot of logic, is that this entire cone is entirely chemical precipitate that's precipitated out of fluids like the ones we've been sampling today. That's 100 meters across, 50 meters tall, if that was really true. It makes you wonder, either it's been as active as it is today for a really, really long time, or else it's even been more active than it is today. There probably is. We've still been trying to work out exactly what the mineralogy is. Well, it could be either one of those two options. This mineralogy, the combination of talc and quartz, when we were here a year ago, that was our best interpretation, was that these were these were actually ultramafic rocks, the things we're looking at right now, and they've had so much fluid flow through them that they've been altered to the point where all that's left is the most resilient mineralogy that's left behind. But what Jill's been doing recently, since last year when we collected the fluids, is actually working out what kind of minerals would precipitate from those fluids if you just generated them at equilibrium and allowed them to cool with seawater. And we know what the range of temperatures are. And if you took the vent fluids from the top of the spire and let them mix with seawater to mix down to the other temperatures we see at the other sites, what you would precipitate out is a combination of talc and quartz. So, it could be the two that, yep, there isn't a good reason why if you were just altering rock, it wouldn't make a nice cone like this. The fact that it's radially sent, the fact that it's radial symmetry on this part of the topography, with a spire sticking out the top, is the one that's probably the strongest argument that it's a chemical precipitate. Yeah, it's built up. It's a bit, the size and shape is actually very similar to the Tag Mound. If you imagine the Tag Mound that hadn't collapsed in on itself, and it rose to a pinnacle. So in fact, this time last year, we still weren't even sure it was ultramafic hosted when we were collecting the fluids or looking at the fluids. The question then had been, we know at Tag that most of the volume of the interior we think is anhydrite, and when Tag goes quiet for a while and dormant, the anhydrite dissolves and it all collapses in on itself. So that was even a third alternate hypothesis in the absence of data, but just some pictures, which is where we were at the end of the Okeanos Explorer, was what if all this mineral was actually anhydrite, because it is a pale colour when you see the fresh broken open surfaces. And we now know that isn't what it is. But that would have been the question of, well, what if this is actually more like a Tag fluid that was basalt hosted? Then we collected the first fluid samples and we knew that couldn't be true, so we did away with that. Then we collected the rock samples and knew it wasn't anhydrite, so that put a second nail in that coffin. But that's the joy of science, right? Testable hypotheses. You come up with constructs of what might be going on, and then you collect the samples to find out if that's right or not. And then when you collect the data, then you go and think again. But that's actually, to get at that, was actually what we were doing on the first leg when we were in AUV mode. What we did was we flew over here with a magnetometer. It took 2 attempts, because that's the interesting thing. You see how Casey's working to fly at this height? Nereus flew without a pilot over this terrain at 25 meters altitude and flew backwards and forwards for a day. I shouldn't tell Casey that. He was not flying at this altitude, but 25 meters off over this kind of terrain is still pretty good going. And the reason we were doing that is from the magnetic anomalies, we can actually, by flying close enough to the seafloor and doing magnetic surveys, we can actually resolve how the magnetic field changes at depth underneath the seabed at the Von Damm Mountain. That'll tell us something about the depth of the circulation of the fluid. No, but he was actually watching us collect the data. We were sending the data to and fro with the joys of telepresence. He actually looked at our first attempt, told us how we could revise the survey and get better data. And so we had a second dive that came back and did more of that. In fact, the same dive that did your photography that you looked at then went on and did the magnetic survey as well. So we're going to do 2 lots of video. We're going to do some video around the vent site first, and then we're going to do Brendan's stuff, so yeah. Because Brendan's stuff is actually on the way up, we can leave the bottom, and oh, of course, we've left the bottom, right? Yeah. Don't worry, we're going to see a lot more shrimp. Okay, so we're approaching the summit of the mound now.
Just on the actual hero. Go to oh, go to oh, I guess it is. Yeah, I was thinking.
So when we're doing... Oh. Okay, so we're moving on again, and we've now come all the way up to 2,316 meters. We're only a few tens of meters away from arriving at the central spire. I'm going to go take a quick look at that, do a few minutes of imaging there, and then we're going to conduct a zooplankton survey on the way up above the seabed. Okay, we are at 4:05. You can tell we're getting close to where we want to be because we are coming in over a sparse shrimp now near the top of the mound. We're down to 36% on our battery power. Okay, let's reset the video while we're getting into the right place then. Yep. Can we do that please, Jeremy? Well, we're not set up at that big hole yet. I would keep going for you, Casey, and we'll get us in the right place. We're not. I mean, we're still 20 meters too deep at the moment, right? 10 to 20 meters.
So do you want me to
So-
stop when I get there or keep flying up over? Are we doing a flyby or a stop?
I'd like to stop and actually look at the orifice,
Yeah.
and we'll do for about 2 or 3 minutes of that, and then we'll do a zooplankton transect up above that. There's two separate things. So we're just restarting our video. For those of you still watching on ashore, we'll ask you to reset your servers or refresh your browser in about 3 minutes time. Is that right, Jeremy? About 3 minutes? And then by the time you get there, if you're lucky, we'll actually be set up in front of the main orifice of the Von Damm spire. We're going to go there, do some high-definition video imagery of the site, and then we're going to do a zooplankton survey up above that on our way back to the sea surface and the ship.